Thank you.
Oh, there we go. Woohoo. All right. Cool. So, hi everybody, and thank you for letting me be here. It's great, even though we can't meet in person. It's still nice to be able to talk to people this way. I run the Invertebrate Behavior and Ecology Lab at the University of Sydney, and that essentially means we study a whole range of creatures, everything from ants to slime molds to bees to velvet worms and soldier flies. I'm not going to talk about all of those creatures today, so apologies if you were here for the bees, because there'll be only a little bit of bee talk. It's going to be mostly ants and slime molds, but feel free to ask questions about any of those species, and I'll be happy to answer them after. If you want to get a hold of me, those are my contact details. If you forget those, my name is pretty unusual, so if you just type Tanya Latty into Google, most of the time you'll be able to find my contact details. So often I think when we think about science, it's the big science we get pictures. And so these are the kinds of things that came up when I just put science into Google Images. These are the big science things, like the Large Hadron Collider, or, banks of genomics and stuff happening. And that's cool. Like I get a bit of science envy sometimes when I see all that fancy equipment, because my equipment is pretty much a net and some Tupperware containers. I don't really do what big science. I like to think of myself as doing small science. Because it's these small creatures, the things we don't normally pay attention to, that, in my opinion, are really running the world. These are, our pollinator species. These are the organisms responsible for, say, cleaning up our waste. They're even pest control. Our whole ecosystem is kind of really resting on the shoulders of these teeny tiny little creatures who've been called the little things that run the world. And so it's really an insect planet that we live on, a planet in which it's things like insects, and it's also non-insects like funguses and bacteria and all these organisms we don't normally think about that are super crucial for everything keeping our planet going. And it's always interesting because when I go into schools and give talks there, I'll often ask kids, what is your favorite organism? And you'll get lots of dogs and cats and dolphins and that sort of thing. But nobody ever really says dung beetles, Nobody ever talks about slime molds as being their favorite organism. And I reckon that's a shame. So my interest professionally, I'm trained as a behavioral ecologist, so I'm really interested in understanding the behavior of organisms. And historically, when we study behavior, we study it in organisms that have big brains. Think of like chimpanzees or parrots or animals that we expect will be able to do all these fancy problem solving. And that kind of makes sense because the vertebrate brain is this amazing piece of evolutionary innovation that allows organisms to take information from all sorts of places and combine it towards making decisions. So it kind of makes sense that most of us who study behavior study organisms that have big brains. The problem, though, is that when you get down to it, the vast majority of organisms on this planet are brainless. And it's always so hard not to throw in politician jokes at this point, but I'm not going to do that. I'm going to be professional. And I'm going to say most organisms on Earth are brainless. And this includes really important animal or organisms like the funguses, like the bacteria, the slime molds, plants. All of these organisms live in a world that is every bit as complicated as ours is. They need to find food, they need to find places to live. In some cases, they even need to find mates. And if you're a fungus and there are 700 different sexes, yeah, dating is definitely a complex sort of scenario. What's interesting about these organisms, though, is that they have to be able to make these sorts of complex decisions in the absence of a brain, in the absence of that sophisticated information processing organ. And so how do they do it? Can they do it? On the other end of the spectrum, though, we also have many organisms that, they have brains, but those brains are small. So think of things like ants and bees, termites, some of the most ecologically important and successful organisms, but they, as individuals, don't really possess a whole lot in the terms of cognitive architecture. And so it's also worth asking, how do they solve problems? And so the answer, so the spoiler here, is that we think of these organisms as solving problems using something called swarm intelligence. And this swarm intelligence emerges when you have independent agents who are not necessarily super clever themselves, but by working together in groups, they're able to achieve problem solving that far exceeds what individuals could do. To some level, this is actually how brains work. So it's strangely enough, brains are also an example of swarm intelligence, because if you think about it, your brain is made up of little neurons. None of those little neurons knows how to write an essay or watch a science talk or do anything really, except for fire. But when you have billions of neurons cooperating together and communicating with one another, you get a brain that can do things that you would never have predicted could emerge if you were only studying the behavior of independent neurons. So in a sense, this is a really, really good example of swarm behavior or swarm intelligence. And it's swarm intelligence that I'm going to be focusing on today in a variety of organisms. So I'm going to start by talking about ant colonies, because I think in a lot of ways ants are probably one of the best examples of swarm intelligence. And then I'm going to talk about a brainless organism, the slime mold amoebas. So if we start with ants, ants are among some of the most diverse animals on the planet. There are around 12,000 species of ants on Earth, although that's almost certainly an underestimate, because every time somebody looks, they find new species. In Australia, we've got around 4,000 species, so we're kind of like a hotspot of ant diversity. And ants are all over the map in terms of what they look like and their behavior, and all aspects of their ecology. Even in terms of sheer numbers of individuals, there is a ridiculous number of ants on the planet. Like, this is an ant planet. That number is the number of ants alive at any moment. It's a 1 with 17 zeros. I think it might be called a quintillion, but it's such a big number, I'm not even sure what the name of that number is. It's a lot. And so we know that ants are, again, in terms of biomass, amongst the most successful organisms on the planet. And while there's a huge diversity in terms of what they look like and what they do, one of the defining features of ants is that they live in groups. So these groups can be relatively small. So the picture on one side of the screen is an acorn, which is only about that big, and inside the acorn is the entire colony of ants. So these colonies rarely have more than 100 individuals. On the other side of the picture is a leafcutter ant colony. And these things can be absolutely enormous. So you can see the humans in there excavating the colony for scale. These structures are really, really big and pretty immense. To give you a sense of just how big these can get, the largest ant supercolony that we know about is found in Europe and spreads over 6,000 kilometers. It contains billions of individuals and has hundreds of different queens who are reproductives. So colonies can get really big, and they can range from being small, they can range to being enormous. But the key thing is that there's always a group of individuals working together in order for it to be a proper ant colony. Oops. So when you have that many individuals, the problem is how you coordinate a society. How do you get the basic things that you need to live done? And in an ant colony, individual ants have brains, so they're not part of this brainless majority, but those brains are tiny. I mean, the average ant has about 250,000 neurons, which might sound like a lot until you realize that humans have 86 billion neurons. So your average ant has a brain smaller than a grain of sand. It's tiny. You might think that, okay, maybe the workers aren't super clever, but maybe the queens or the reproductives are the brainy ants. And that's not true either. A queen ant isn't really a queen in the sense that she controls the colony's behaviors, really. She's really just the reproductive parts of the colony. So her job begins and essentially ends at producing more ants. She's no smarter than any of the other ants in the colony. So in a really real sense, in an ant colony, you've got lots of individuals, nobody's very smart, nobody's in charge, yet somehow the colony manages to work. And to give you a sense of how sophisticated these societies can be, ant colonies evolved agriculture millions of years before humans did. So this is a picture of an ant, and you can see it's doing something strange with that aphid, or that aphid-like creature in the corner. What that insect is doing is it's drinking plant sap and pooping out sugar water. And the ants are harvesting that sugar water and using it as the main energy source for the whole colony. So the ants are essentially treating those little sap-sucking insects in much the same way humans treat cows, which is kind of astonishing. But more than that, the ants actually will protect their little herds from predators. So if any predator comes by, they'll fight it off. They'll move the aphid or the sap-sucking insect to a new plant if it needs to be somewhere better. And in some species, queen ants who are going off to start their own colony will carry a tiny little baby aphid under their wing, and that will become the starting stock of their new colony. So it's incredible. It's like livestock agriculture. But you want to go a step further. Leafcutter ants are found throughout South and Central America, where they are very well known for cutting out chunks of leaves and carrying those around the forest. But they don't actually eat the leaves. What they do is they mash those leaves into a pulp, and then they use that pulp to grow a fungus, and it's the fungus that they eat. So they're like mushroom farmers. The ants as a colony are very good at selecting the right types of plants that maximize fungal growth. They will weed the garden, so if the wrong type of fungus starts to grow, they'll cut it out and throw it into the waste bin. Scientists even noticed that some of these ants had this weird white coloration across their exoskeletons. And at first they thought maybe those ants were sick, maybe they were infected with something. But it turns out that those ants are growing a bacteria specifically on regions of their exoskeleton. They feed that bacteria to keep it alive. And in response, that bacteria secretes an antibiotic so that when the fungus garden gets infected with pathogens, those ants can rub themselves against the fungus garden, transfer that antibiotic, and kill the pathogen. So in a sense, they've even evolved a form of pest control as well, which is astonishing. Another example that I love, a little bit closer to home, these are green tree ants. They're found mostly up north, so sort of around Cairns area. And they're often in trees where they will build their nests by combining leaves and sewing them together with silk. So you can see a little bit more closely here. Now the process of building these nests is astonishing because each individual ant has to pull on the leaf to curl the leaf just in exactly the right position. And then adult ants don't produce any silk. So to sew the leaves together, they have to use their offspring. So that's an ant holding a little baby ant, a larva. Larvae produce silk, which they use to make their pupa when they eventually pupate. And so the adult will just squeeze that larva gently. The larva in response will produce silk, and they'll use that silk to sew the nest together. And again, remember, these insects have tiny brains. So no individual involved in this process is really aware of the overall architectural plan. But with lots of little individuals, each making simple decisions, they're able to create these really beautiful and functional nests. So I think one of the best examples I can think of ant swarm intelligence, if you will, has to do with their transportation systems. I like to talk about transportation systems because I think it's something we can all relate to. Almost every human civilization has had some form of transportation system, and these systems are absolutely crucial because they allow us to move individuals from place to place. They allow us to deal with, to move resources, even to spread things like ideas and innovations. So building a good transportation network is often at the very backbone of any human society. And one of the questions you have to face when you're trying to build a transportation system is where to put the roads. So if you imagine you have a whole bunch of points, let's say these are, I don't know, cities in a country, and you needed to connect them up using road or trail or track or whatever, you can ask yourself, what is the best way to do that? If you think about it a little bit, you start to realize that there's probably more than one solution because it depends on what you're trying to optimize. So one possibility might be that we want to connect up those points using the least amount of trail. Let's imagine that building trail costs a certain amount of money. We want to minimize the money we spend. We could do that using something called a minimal spanning tree, which is a way of connecting points that's really low cost. So that's kind of a handy feature. Unfortunately, the problem with minimal spanning trees is that although they're cheap, they also have basically no robustness. So, if anything happens to any part of that trail, at least one of those points will become disconnected. There's only one way to get through that network, so it's not particularly robust. It's very easy for disruptions to cause problems. So we might say, okay, I don't want a fragile network. I don't care about cost. I want the most robust network possible. This Delaunay triangulation, borrowing words from graph theory, this is another way of connecting up points that makes a really high robust network. And that's cool because now you can have all sorts of damage to all these trails, and it still takes a lot of damage before at least one part of the network becomes disconnected. That's good. But as I'm sure you've noticed, the downside is it's really, really expensive. So this idea forms something called the transportation problem that was formalized in the 1700s, but I suspect people have known about probably as long as they've been building networks. which is that you can build a network that's really robust, that has lots of connections and it's very difficult to break, or you can build one that's really cheap, but you can't do both at the same time. So in practice, human engineers work really hard often to try to find somewhere in the balance point where it's like robust-ish and cheap-ish, but it's not optimized on either count. And given how sophisticated this sort of problem is, I mean, there's no equation that you can use to solve this problem. I was really curious to see what ants did. Because ants, just like humans, have to build transportation systems. Often they use that to connect their nests to food sources or to their little herds of aphids or to the trees they're harvesting for their fungus gardens. Some species also use those trails to connect up nests. So a colony will actually consist of many independent nests, or sorry, not independent, many interdependent nests that are themselves connected up with trails. You can think of them almost like cities in a country. They're all related, they're all interacting, and they're connected by trails. And so today I want to tell you about two ant species, two very different ants in their ecology and their behavior. I'm going to tell you how these two species negotiate this transportation problem. So if we start with the first one, this one's the Argentine ant. Argentine ants are super invasive. They're not native to Australia. They're amongst the world's 100 most invasive species. So in that picture, the Argentine ant's not that big ant. It's the little ant. It's those tiny little ants that are beating up the bigger ant. When Argentine ants move into a region, they typically outcompete most of the ant species that were there before. So they can be kind of nasty that way. For my purpose is what's interesting about them is that they do have this thing called polydomy, which means they have many, many, many nests, and those nests are all connected up with trails. So we can use that to start asking questions about the types of transportation network an ant might build. And again, small science, I don't have a large Hadron Collider. I have a black pond from Bunnings that I painted white and drilled some holes into it. And so the ant colonies are actually sitting towards the bottom here, at the bottom of the, just sort of down here underneath the Bunnings container. And the ants can crawl into the container right here. These are the little holes that we've drilled in. So the ants can climb up from underneath the container into the arena, and then they can build their nest network however they want by connecting up those points. So in this case, we've got 3 points where those sticks are that make up the places they might want to connect their link to the nests. Oops, right there. Okay, so you can ask yourself, if you had 3 points, so this is the configuration, one of the configurations we asked the ants to connect, how would you connect those up? So to start with something easy, imagine you were going to do the shortest path between those 3 points. What would it look like? So I'm going to give you a few seconds just to think about what that structure would look like if you had to connect up those 3 points. If you said this, then congratulations, that's a Steiner tree. This is the shortest way to connect up these 2 points if you're allowed to use nodes, so that little node in the middle there. There's also something called a minimal spanning tree, which would be kind of making a V-shaped, which would be the second shortest way to connect up those paths. So, oops, sorry. There we go. Oops, sorry about that. I seem to have unshared somehow. There we go. All right, so that's the shortest way. And this is a picture of the actual experiments. This is what the ants did when I gave them this problem. So again, the red arrows are showing you where the nests are located. So the ants have climbed up into the arena. And that sort of squiggly gray line is the ant trail. And what you can see is that they've done a really good job of connecting these up using what looks pretty much like the Steiner tree, like the shortest path solution. Now, in case there's any mathematicians in the audience, I'm going to say it's like a Steiner solution because technically to make a Steiner tree, the lines need to be straight and they need to hit at exactly the right angle, but they're ants, right? I don't expect them to move in perfectly straight lines, and the fact that they've come up with something that's pretty close, I think is astonishing, especially when you consider that they could have done anything. You could have like a cobweb of points, you could have all sorts of extra loops and things, yet you put all these ants together and they're connecting up their nests using the shortest path, which is a solution that's not even easy for humans to do, yet these teeny tiny ants are able to do it as a colony. And just in case you think I'm only showing you the one time it happened, here's just some other pictures from that experiment. Most of them are Steiner-like trees. Again, they're not perfect, but they're ants. The one in the top left corner, which looks like a V, we also saw some of those, which are not Steiner trees, but which are the second best way to solve that problem. And the ants will consistently do this far more often than we would expect from chance if they were just randomly connecting up points. And those are just to show you where those are a little bit more clearly. So what's exciting is that we actually have an idea how they do this, because we know that individual ants, I mean, I love Argentine ants from a research perspective. They are not smart. Even amongst ants, they're not clever. I mean, these animals are astonishing. They pretty much just walk in circles if you leave them by themselves. But we kind of have an idea how they work together as a group. So as an Argentine ant is walking, she's constantly tapping her abdomen against the ground. And as she taps, she's laying this tiny drop of an attractive chemical called a pheromone. We know that other ants are more likely to walk in places that have pheromone than areas that don't, and we know that they're more likely to go to places that have higher concentration of pheromone than places that have low concentration of pheromone. The other key piece is that pheromone evaporates over about a 20-minute period. So the longer it's left without being reinforced, the faster it evaporates. And so what happens is this. This is a time lapse of the arena. And at first you can kind of see that you just have ants pretty much everywhere. Every one of those little gray dots is basically an ant. They're just kind of a mess. As time goes on, they start to connect up those the nests. You can see that happening in a few places, but it's not a great network. Like there's weird loops. There's a trail that seems to be going off to the right. I don't know why they're going there. There's no food, there's no nest. It's just kind of a mess. There's all sorts of stuff. But over time, the most direct way to get between the 2 points, those will get more ant passes over time. And that's simply because they're short and direct. So there's a higher density of ants walking on those than an ant on a long meandering trail going nowhere. Because the ants are more attracted to areas of high concentration than low concentration, then that higher density of ants translates to a higher concentration, which attracts more ants, which leads to a higher concentrations, which kind of sets off this almost snowball effect that causes the whole network to start to organize itself. And you can see that happening here, that the long meandering trails or the loops are starting to disappear, and the short direct trails are getting reinforced. So by the end of this, you end up with something that looks pretty much like the minimal spanning solution. This is the minimal spanning tree. So not the Steiner solution, but the second best. Pretty fantastic. And we suspect this is really how ants are able to optimize their networks, using this relatively simple way of working. But if you recall, so I think this is pretty cool, but if you remember what I said earlier about how minimal topologies are typically not robust, while the ants have done something that's very cheap, it's not a great network if you're worried about resilience, because any damage anywhere to any of those trails and one of those nests is going to be isolated. But we think that it doesn't matter so much potentially for Argentine ants, because ant networks are basically self-healing in this species. So if you ever see like a trail of ants outside and you say put your foot, in front of them, they kind of just flow around obstacles like water. And because they're always laying pheromone, it can take them seconds to get around a barrier. They're able to heal their network really fast. And so we reasoned that perhaps that ability to self-heal the network means that they haven't really evolved these mechanisms to build topologically robust networks. But like I said earlier, there are a lot of ants, and different ants have totally different ways of building networks, including some species that, unlike the Argentine ants, actually invest a lot in constructing physical networks, either by tunneling, or in the case of my favorite ant, the Australian meat ant, by physically clearing all the vegetation from their path. So the little picture that looks like a star in the corner, that's an example of a meat ant network where the ants have physically chopped every single bit of vegetation to create these kind of ant superhighways. These can be so incredibly obvious that you can even see them on Google Earth. So the picture on the top left, that's from Google Earth, and you can just barely see in the corner these little meat ant networks that look a little bit like neurons or stars. That's how clear they are. So because the meat ants are investing so much energy in clearing their paths, we reasoned that they might build networks that had more robustness, that might be more at that balance point. Because you can imagine if a tree falls across a meat ant network, they can't fix it as quickly as the Argentine ants can. It would take them quite a lot of time to build a way around that obstruction, and in that time they're losing efficiency. So it would make sense to build more extra routes to kind of be able to get around in the event of damage. So that's just a close-up of a meat ant network. So what we did is we went out and we mapped 110 meat ant networks, which is no small task. And then we looked at what the topology of our networks were, the meat ant ones, and we compared them to networks that had known properties. So I'll give you an example. Here, the blue circles would be the location of meat ant nests, because like the Argentine ants, meat ants have several different nests that they connect up with trails. So blue dots are nests, the lines are trails. If the meat ants were connecting up their nests using the shortest possible path, you could imagine a topology like the one you can see here. On the other hand, if they were building really robust but expensive nests, we'd expect topologies that look like the one on the far side of the screen. And in fact, what we saw most of the time were nests that kind of look like that one in the middle. Networks that were balanced pretty much at that balance point. And again, we looked at 110 networks, and there's sort of a simplification. We did lots of various comparisons to look at all sorts of features, and the takeaway message was that meat ants, by and large, kind of sit in that middle zone. So they're not too robust, not too expensive, but also not too fragile. And that makes sense given what we know about their ecology. So what's neat about this, I think, is that we've taken two different species of ant. They're solving the same relatively complicated problem, but coming up with different solutions that reflect their particular ecological needs. And that's only two species. Like I said, there's so many more ants that we could look at. It'd be fascinating to see how they're able to solve these problems despite having teeny tiny little brains. So that's just to give you a taste of how ants solve problems. But remember, ants, they have brains. So it's still the case that you have lots and lots of little tiny brains coordinating. But what if we went to the next level and thought about that brainless majority, those organisms that don't even have brains, full stop? So without further ado, I'd like to introduce you to my favorite brainless organism. These are the acellular slime molds. Acellular slime molds have some pretty funny common names. So the yellow one up there is called the witch's butter slime mold. The one in the middle, that brownish one, is often called the dog's vomit slime mold. There's even one called the wolf's milk slime mold. So not the most attractive names, but they kind of reflect what slime molds really are, which is gigantic blobs of moving mucus, basically. Even though they're called slime molds, they're not actually funguses or fungi. They belong instead to the kingdom Protista. So the kingdom Protista is a kingdom of life that contains single-celled organisms that we really just don't know where else to put. So they're not animals, they're not plants, they're not fungi, they're one cell. We'll just call them protists. The closest thing a slime mold is related to is an amoeba. So it's essentially, you can think of it as just being a really, really gigantic amoeba. And when I say big, I mean big. These things can get pretty large. Some of them, the largest one I've ever heard of was a meter, more than a meter long. And I'll show you a bit of a time-lapse video. This is what slime molds look like in the wild. Now this is sped up, but in real life, slime molds can move at about 5 centimeters per hour, which is pretty fast when you consider you're dealing with something that is basically just a giant blob. All the yellow goo you can see in that particular pic in this video is one enormous cell. So even though it's one cell, it contains millions of nuclei, which means that you can cut that cell into as many pieces as you want, and it'll very happily go off. Each of those pieces will very happily go off and form their own individual within minutes of being cut off from the main cell. If you put those bits back together again, they quite happily rejoin. So here's two cut pieces of a slime mold, and you can see they're merging back together to create one slime mold, happily reforming. In the wild, slime molds kind of ooze around the forest floor, engulfing anything they can get their pseudopods on, bacteria, mushrooms, whatever they can get. Oops. So this is basically the blob. If you've seen the movie The Blob, you pretty much know what a slime mold is. It's this big mucusy thing that kind of oozes around the forest floor just eating stuff. But remember, because it's unicellular, this organism has no brain, it has no organs, it has no neurons. It has nothing similar to what animals have in terms of information processing, which I think makes it a really cool creature. Now, back in 2000, this scientist, this is Dr. Toshiyuki Nakagaki. He's at the University of Hokkaido, and he's a biophysicist. So he was really interested in studying how slime molds move, because the slime mold is a great organism to study to understand how cells move because they're big. So you can really see what's going on, much more so than you can in most single-celled creatures. So that was Toshi's main job. But then one day, and I've asked him what made him think of this. He's never really told me. I don't actually know. But one morning he woke up and he had what I think is probably one of the weirdest ideas in science. He said, what would happen if I took this giant mucousy blob and I put it into a maze? So, I mean, that's odd. I mean, we use mazes to study the behavior of brained organisms. And, when you first put it in the maze, at first it kind of does what you would expect, which is nothing. It kind of oozes around, fills the maze. Not much happens. The two white things at the bottom of your screen, those are two pieces of food. So the slime mold has kind of filled the maze, not much happens. But if you watch closely, you're going to start to see some structure emerging. So the slime mold is going to start to move away from some parts of the maze. It's going to move away from parts, for example, that are dead ends. And it's also going to move away from loops. And so it's slowly retracting its biomass, so that it's no longer filling the maze. And if you give the slime mold between 48 and 72 hours, most of the time they will find the shortest path through the maze. Now I think this is astonishing because, I thought I was edgy studying animals with small brains. This thing has no brain at all, and yet here it is solving a shortest path problem, a maze problem. And that's incredible. And I was fascinated when I heard about this the first time. But not to be outdone, about 10 years later, the same research group did a second experiment, except this time what they did is they got a map of the Tokyo metro system, and they put little dots of food at the location of the major stations on the Tokyo metro system. And then they let the slime mold do what it does and slime around. The network that remained at the end was almost identical to the real Tokyo metro system. And this has become kind of a fun game that slime mold researchers just play in whatever country you happen to be in. You see, is your network as good as the slime molds? The reason you get such similar networks between human engineers and slime mold amoebas is just that they're trying to do similar things. Both are trying to kind of find that balance point between robustness and cost, and slime molds are able to do that almost as well as people are, but no brain at all. So to me, that was fascinating. And I got really excited about this, as did a lot of the media. So there's every few years, there's papers that come out about what slime molds can do. And you get stuff like slime molds solving Brexit and can slime molds predict elections? All pretty interesting stuff. Now, for me, as I said before, I studied behavior and ecology. And when I first started working with slime molds, my main interest was understanding how individuals make choices. And so while I thought it was really cool that slime molds can solve shortest path problems, I started to wonder if that was all they could do. And I feel like a terrible person saying that because, the fact that this brainless amoeba can solve mazes is pretty astonishing on its own. It doesn't need to do more to be cool. But as a behavioral ecologist, I wanted to know whether, has this organism evolved only to be able to solve these shortest path problems, or can it do more? Does it have a bigger behavioral repertoire? Can it do more things? And so, like I said, I was really interested and still am in how we make choices. Decision making is hard, and if like me, you've been kind of stuck at home for the last couple of months doing probably too much internet shopping, my big question was, can I find a really nice insect-themed dress to wear? Lots of choices because the internet is awesome. But decision making can be quite tricky. Choosing one of those dresses or one of whatever is difficult because there's usually more than one thing we care about. So if I asked you whether you wanted $1 or $1 million, that's easy, right? You choose the $1 million because more is better than less. There's only one attribute there. In reality, though, life choices are full of many, many aspects. Even choosing between coffee makers involves a huge list of features. Some things will have the features I want, some won't, but I have to do something in my head to kind of put all of those together towards making a decision. So to give you another example of why this can be difficult, I was really lucky a few months ago before before the pandemic hit to have a sabbatical back home in Canada. So if you were trying to guess my accent, it's a Canadian one. And while I was there, I visited Montreal, where I was able to have some of the, well, I'd say Canada's premier dish. For those of you that are unfamiliar with poutine, it's thick cut chips with melted cheese curds slathered in gravy. This is Canada's single most important innovation to the world. It's a gift to the world that we gave you. And it is absolutely, indisputably awesome. It's amazing. It's delicious. This is a particular variety where they've used sweet potatoes instead of regular potatoes. Oh, great. But as awesome as poutine is, eating it will kill you dead. And so when you walk into a restaurant, there's usually another option. So let's say salad. I love salad as much as the next person, but it is demonstrably, objectively less awesome than poutine. Nevertheless, when I walk into a restaurant, it's a little bit of a hard decision because on the one hand, I care about how awesome my food is. On the other hand, I want to live to see my 50th birthday, and that's not going to happen if I eat a lot of poutine. So I have to weigh up those two different things, the health consequences of my food choice, but also the just sheer awesomeness. Now, other organisms have to make these choices too. It may not be healthy and awesome. It can be the risk of getting eaten by something versus the quality of the food or any number of things. And so it's a decision we see across the animal world. Now, the easiest way to solve this kind of choice is to simply focus on one attribute only. So maybe I always choose the healthiest food, no matter what the options are. Maybe I always choose the awesomest. The important thing is that this allows me to make a decision quite quickly because I basically throw out most of the information and I focus only on one attribute. Because this is a cognitively simple task or way of solving the task, this is what we would expect simple organisms that didn't have big brains, we would expect them to do this. If we're a little bit more sophisticated though, what I could do is consider both awesomeness and health and then do some sort of mental calculation to choose the one that gives me the best trade-off between those two. So in practice, what this would be is that I've got some internal calculation about the health value of a food. I have some internal scoring system for the awesomeness. I do a calculation in my head, and then I choose the one that has the biggest score. Now, no one's saying that animals or humans are doing this consciously, like you're not consciously putting on numbers to things, but an approximation of this process is what we expect humans and other more sophisticated animals to do. The nice thing about this kind of decision-making strategy is it allows me to make trade-offs. So instead of always choosing the healthiest or always choosing the most awesome, even when the difference is small, I can make trade-offs between those. So to give you another example, again from Canada, I don't know what happened to Canadian cuisine while I was away, but this thing, it just came out when I arrived. It's a maple sweetened Belgian waffle with a sausage patty, cheese, and I think some kind of weird egg, all sandwiched together. It's awesome-ish, mostly for the novelty factor, but it's also terrible health-wise. But if I walked into that restaurant, I had a choice between the waffle monstrosity and the salad, I could still do that mental calculation and calculate that although the waffle is slightly more awesome than the salad, the exceedingly high health value of the salad will kind of tip the scales, and now I'll choose the healthiest. So I can be a lot more flexible in my decision making if I can do these kinds of mental calculations. However, we didn't really expect to see that in organisms that have smaller brains, or indeed organisms that have no brains. So one of the early experiments we did was to see how slime molds deal with this kind of a trade-off. And so to do this, we took advantage of two factors of slime mold biology. One, slime molds love oatmeal. I don't know why, they don't get it in the wild. It's just a weird quirk. But what that means is that we can mix oatmeal with different amounts. We can make food discs that have different amounts of oatmeal. So really concentrated ones that are really delicious for a slime mold versus ones that have less oatmeal and are less delicious. The second feature of slime mold biology we take advantage of is the fact that slime molds do not like light. Light is terrible to them. They'll always try to find dark areas over light ones. I'm actually convinced that the fact that they're scared of light is the only reason they haven't taken over the world at this point. And what that means is that we can set up decisions for the slime mold where it needs to choose between a really high concentration oatmeal disc that's in the light and thus scary versus a low concentration food disc that's in the dark and thus safe. Now, we can then combine different ratios between the concentration of the low and high disc. So again, if the slime mold is doing the simple thing, we might expect that it will always choose the high concentration food no matter whether it's in the light or dark, or the converse, that it'll always choose the safe option irrespective of how much better the option in the light is. And if you're wondering what a slime mold looks like while it's making a decision, here's an example from a slightly different experiment. In this case, the good quality food is at the very top of the screen. And so you can see that after an hour or so, the slime mold will spread itself across all food discs. This isn't growth. So the slime mold isn't growing over this time frame. It's just redistributing its biomass. But then over time, you can already see it starting to push more biomass towards the top food disc, and it's starting to pull away from the options it likes least. And again, if you give the slime mold around about 5 to 6 hours, depending on what kind of mood it's in, most of the time the slime mold will end up on one of those 3 food discs. And in this case, it made the right choice, and it's chose the option that's the best, that has the highest concentration. So it's relatively easy to set up these experiments and then just come back and photograph where the slime mold is and which option it ultimately chose. And so we did this by setting up, again, combinations of food concentration and light. And what we found is that, so what you're looking at here on the vertical axis, that's the number of slime molds that ended up in the light. So the ones that said, okay, I'm going to go hang out in the light, I don't really care. And then on the horizontal axis, what you can see is the concentration difference between the one in the light and the one in the dark. And so when the concentration difference between the option in the light and the option in the dark is relatively small, so the dangerous option is only a little bit better, most slime molds typically stayed in the dark, which kind of makes sense. They chose the safe option, even though they were losing a little bit of food. On the other hand, when the option in the light was much better, so 5 times or more better than the option in the dark, all the slime molds went into the light. So, and then there's sort of this region in between where some went into the dark and some didn't. And so what that tells us is that the slime mold somehow is able to make those kinds of trade-offs. In order to do this, it must be keeping track not only is the option in the light or not, but also what's the concentration difference between my 2 choices. It's calculating something somehow and then making the decision that works out to be the best trade-off between those 2. That's astonishing because remember, this organism has no brain. We have no, it has no neurons. There's nothing that's similar at all to what we would expect from brained organisms, yet it's doing something that we consider a fairly sophisticated form of choice behavior. And, you know, I could spend 3 talks talking about all the cool things slime molds can do, because the more we study them, the more we find that they are behaving much like we would expect animals to. They're able to change their behavior, their search pattern, depending on what they just ate to maximize the chances they run into something good. They're able to habituate to negative stimulus. They're even able to balance them, to spread themselves across a whole bunch of foods in the precise way they need to in order to make sure they get the right balance of protein and carbohydrates. And these are just a small sample of the things they can do, yet they're doing it all without a brain. So how is that possible? And so I felt I should mark this part of the talk as it's kind of speculation. The truth is we're still trying to figure this out, but we do have some ideas. So I'm going to show this again. I don't know if it's coming, it'll come through quite right on Zoom, but we'll try. And then remember, this is where the 2 slime molds were coming together. But I want you to pay attention to the fact that the slime mold appears to be pulsing. Now, if you can't see that, that's okay. I'll just sort of describe it to you. But what's happening is you have a big yellow slime mold, and some regions of the slime mold are pulsing at different rates. And you kind of see almost waves of pulsing spreading through the slime. Now, we know that regions that are in contact with something that it likes will pulse faster than regions that are in contact with something the slime mold doesn't like. We also know that increased pulsing causes more goo to flow in that area, and that goo causes the tubes to expand. And so you get more goo flowing through the tubes, which causes more expansion, which causes more goo. And we think, oh, right, and each of those pulsing regions we know can entrain or kind of communicate with the regions around it, so that if you're a pulsing region close to a very fast pulsing region, you'll start to speed up. So what we think is happening is that some combination of all these little pulsing bits kind of collectively allows the slime mold to do all of these, pretty incredible things. So what I think is fascinating is that this seems very similar to what we see in a brain. So when some of the theories behind how brains make decisions, imagine that each population of neurons essentially votes for which option you want to look at. And so some neurons will be voting for one option, others will be firing for another. By vote, I mean the firing rate. and that collectively that's how you make a choice. That's also very similar to what ants do. So when ants are choosing between foods, they're depositing little droplets of pheromone. That's sort of equivalent to them voting for one option or another. And again, you get these feedback mechanisms that allow them to focus on one choice. And again, the slime molds, you get the same sort of thing, different pulsing regions. So we're starting to see these similarities between brains, swarms of ants and slime molds that suggest that although these things are running algorithms on very different hardware, maybe that underlying decision-making algorithm isn't all that different between systems. It's speculation at this point, but it's sort of an interesting idea to think that there may be these deep commonalities between all of these different systems. Okay, so, So, the takeaway message here, I guess, is that even if you don't have a brain, or even if you have a very tiny brain, swarm intelligence or collective behavior can still allow organisms to solve complex problems without needing a brain. Some said brains are kind of overrated. There's lots of workarounds that I think we as behavioral ecologists may not have been paying as much attention to, because we've often been thinking that you need to have a big brain in order to do complicated things. It's becoming much more clear that that is not at all the case. But so what? I mean, it's interesting intellectually, and I think that's important too, just to study things sometimes because they're cool and because we find them fascinating. But, I have grants to apply for as well. I think it is useful to think about what's the point, what's the purpose, like what can we do with this information? And I think this information is important for a couple reasons. I work quite a lot with engineers and mathematicians and computer scientists in the field of bioinspiration. And so this is the idea that maybe we can learn from nature towards building better technological systems. Now, if you think about things like the Internet of Things or autonomous vehicles or swarms of drones, a lot of our technological systems are becoming more complex and more decentralized. We're moving away from a model where we have one central controller that's controlling all of our technology, and we're moving more to like the internet sort of style of technology. I would argue that if we're going to move in that direction, why not start looking towards systems that do that well, things like ants and slime molds, because they have evolved really good mechanisms to run decentralized systems. And they've been shaped by millions of years of evolution, whereas humans, I think we're much more hierarchical. We don't really think about things in a decentralized sort of way. So it makes sense to look to these systems for inspiration. And we're starting to see ideas like swarm robotics. And I think this is a really cool idea because one of the problems you have with, say, space exploration is that you might spend billions of dollars making one rover, but if that rover crashes, that's it. Like, it's over. And same whenever you're in a hostile situation, so search and rescue after natural disasters, for example. The nice thing about swarm robotics is that you can have lots of relatively cheap individual robots that collectively solve problems, but if you lose a few, or half of them even, each of them is still able to function together as a collective. So these are very resilient systems. And there's been talk even of using these kinds of swarms in agriculture as well. So that's one of the potential uses. And I always like to bring this up in talks because I think sometimes we forget that we're in the middle of this incredibly gigantic mass extinction event where we're losing species at an appalling rate. We're losing species so fast that many of them are disappearing before we even give them names. And while I think there's all sorts of ethical and ecological reasons why that's terrible, one of the reasons we don't often think about is the fact that each of those organisms has been shaped by millions of years of evolution to have solutions to problems we have now. And every time we lose one of those species, we're losing that knowledge and that potential for inspiration. It's like burning a library without knowing what books you're burning, without even knowing the titles of those books. It's completely terrible. And I think sometimes we forget about that when we talk about bioinspiration, that we need to harness that diversity of nature if we're going to start solving some problems. And so along similar lines, I think understanding collective behavior and swarm intelligence can also help us with conservation, because many organisms, and many of which are ecologically pretty important, like honeybees and stingless bees, they live in colonies. And when we talk about colony collapse disorder, what we're really talking about is the failure of a swarm intelligence system. It's a system, a collapse of a society. And so if we can understand better why that happens, we can maybe start to fix it. And so one of the things my team has been working on quite a lot is trying to understand the mathematics behind colony collapse to try to get better ideas about how we could stop that from happening. I think in the agricultural space, understanding how swarms work is also really important because we can start to harness some of that power towards building more sustainable agriculture. So we know that ants are really, really good at finding and killing and eating pest insects, but we're still at the very early days of figuring out how we can use that towards better biological control. And I'd argue that understanding how collectives work can help us understand how we can direct that to our benefit. So I guess the takeaway message here is that nature is diverse. There are so many cool and interesting organisms that I didn't even have time to tell you about, like the velvet worms and the soldier flies. And I think keeping these organisms around is super important. So conservation, it can't be overstated how amazing the biological diversity we have. We're super lucky to live on a planet that's full of nature that we can learn from. So let's try to keep it that way. So that's all that I have to say. Should I start trying to answer some of the questions? Thank you all for listening to me ramble on about, some cool critters.
Thank you. Thank you so much, Tanya. That was an incredible talk. Wonderful. Yeah. You give yourself a chance to draw a breath and have a sip. And whenever you're ready, sure, feel free to dive in.
Cool.
We've got 4 questions already. And if anyone has any they'd like to ask, just type them into the Q&A box, please.
Okay.
The question and answer box there, Tanya?
Yep, I got it. So the first one from Daniel is, does warmer conditions, including fire, speed up or trigger entomological behavior, including egg hatching? And does that influence bird nesting behavior? Okay, so the first part of that, insects and indeed most invertebrates are, we call them ectotherms. So it means they're not able to regulate their own body temperature. They depend on the temperature around them. So when it gets warmer, they tend to get faster. They're able to do more. They're more active. So yes, heat, climate change and warming will change the behavior of insects. Whether that's a good thing or a bad thing is kind of all over the shop. There are a lot of negatives to increased temperature, obviously. When we think about things like pollinators, increased temperature can cause flowers to bloom earlier than they normally would, which means their pollinators are no longer out at the right time. So there's all sorts of interactions between temperature and behavior that I don't think we really understand all that well. My research group is starting to do a little bit of work on the impact of extreme temperatures on swarm intelligence and colony function in honeybees and to a lesser extent stingless bees. So we're trying to get a better understanding of how temperature influences all of these things. But I mean, at this point, I think we don't really know a lot of the details. As for how it affects bird nesting, I'm gonna tap out of that question just because I don't really know much about birds. So I can't answer that one, I'm afraid. What conditions in this season cycle encourage more slimels to procreate? Well, okay, so first of all, slimal procreation is completely complicated. So if we start from the form that I showed you, which was like that kind of creeping blobby creature, that's one phase of the life cycle. Now, it can stay like that for a long time, but if conditions get bad, so it gets stressed out, it can form a spore. And that spore almost looks like a teeny tiny adorable mushroom. And that's actually why people thought they were funguses for a long time. Now, inside of that spore head are thousands of little spores. Those get dispersed either on the wind or by sticking onto other animals. And if they get moist, they can crack open and you get this little thing that looks like an amoeba that climbs out. But it's haploid. So by haploid, I mean it has half the complement of chromosomes. So it's kind of like almost like a sperm or an egg cell, except that it can eat and multiply and do its own thing quite happily, for essentially indefinitely. Now, if that little haploid amoeba runs into another haploid amoeba of the right mating type, and for some slimels there can be 700 different mating types, this one has to find the one that's exactly the right mating type for it, then they'll mate, then they'll form a diploid amoeba, so now with a normal number of chromosomes, that that diploid amoeba will have one nuclei, and that nuclei will divide over and over again, but it won't leave the cell membrane. And so that blobby thing, the plasmodium, is what happens, and then we're back at the beginning of the cycle. So it's very complicated. As far as what encourages it to do that, it depends on which life stage you're talking about. So slimal plasmodium, that blobby crawling bit, that tends to happen more in like damp kind of conditions. It's not very good at being dry, and usually darker conditions for that to happen. Same for the spores to crack. They usually need some water for that to happen. Yeah, so there's a whole bunch of different conditions. Mostly cool and damp, though, would have been the shorter way to answer that question, perhaps. Where do ants go in cold weather? And why are they not visibly active in cold weather? So same as what I'd said before. Cold weather just slows insects down. So in climates like I'm in Sydney now and it's cold, like there's still ants out, but they're just not as active. There's not as many of them. And that's just because it's harder for them to eat. It's harder for them to move in the cold because their metabolism slows right down. Slime molds, how toxic are slime molds to humans? Slime molds are not toxic to humans as far as I know. I've never eaten one, so it's hard to say. I've never heard of it. I've only ever seen one medical account of a person being infected by a slime mold, and it was a person who was severely immune compromised who cut themselves and got slime mold in the wound. Generally speaking, they're classed as like the least harmful class of microorganisms, so they're not going to make you sick, pretty much. Can I get a slime mold as a pet? Strangely enough, I started working with slime molds because I had one as a pet. So I was working on ants, and I visited a lab in Japan and saw their slime molds. And I came back to Australia and Googled, can I get a slime mold as a pet? Turns out you can buy slime molds online, and that's how I got my first slime mold. I just kept it in a drawer in my desk in a little Petri dish, and I would feed it oat flakes. And because I was working on behavior of ants, I started noticing that when I fed the slime mold, the patterns that it made looked just like the patterns of trails I saw when I was feeding my ants. And that's how our first experiment started. So yes, you can keep slime molds as a pet. I probably wouldn't recommend it because they grow annoyingly fast and they tend to escape from your dishes. So you end up with slime mold crawling on your walls. I'm not allowed to keep them at home anymore because of previous incidents with escaping slime mold. So they're fun and you can totally get them online. They're also super easy to find in the wild. So yeah, I mean, if you're interested, go ahead. Just Google it and you'll be able to find them. Is there an opportunity for volunteers to perform experiments for me? Yeah, sure. We do take volunteers in the lab. Right now, everything is a little bit up in the air because of pandemic safety stuff. But if you are interested in volunteering, just drop me an email and we can see if we can set something up. So it's tanya.latty@sydney.edu.au. If you're not sure, just Google Tanya Latty and you should be able to find me. This talk was fascinating. Can you say something about velvet worms? Oh, yes, thank you so much for asking that question. The initial version had velvet worms in here, but then I realized I was going to be talking for hours and nobody probably wanted that. You can see velvet worms on my screen right now in the far right corner. That's a little velvet worm. They're these amazing animals that are not insects, even though some people think they are. They're an ancient, I'd call them a sister species to insects. So they're almost like a fossil taxa. They haven't changed much in millions of years. They've got these cute little stubby legs, and the coolest thing about them is how they hunt. So slime molds have little glands underneath their tentacles that shoot glue. So they'll find their prey, shoot glue at it. The prey becomes immobilized and it can't move, and the velvet worm will crawl over it, slice it open, inject it with venom, and then kind of eat all the goo inside. So they're fascinating creatures. We've started studying a few of them in the wild because, surprisingly, some species are actually social. So you see groups of them living together, and what we were interested in is trying to figure out were they hunting in groups? What are the dynamics of those social groups to try to understand more about how the species worked? Unfortunately, the area that we collected them in has subsequently burned down during the fires pretty badly. We were hoping to go back and see the extent to which the population's been affected, but because of COVID, we haven't been able to. So right now we have 200 velvet worms in the lab, and we're unsure about, are they the last members of that population? Did the ones in the field survive because they were inside logs? We don't know. So it's kind of, I guess, brought home how important conservation is, at least to me, because I never thought I'd be on the side of having a bunch of organisms in the lab that may be the last of their kind. We don't know. So it's a bit grim. There are lots of species of velvet worms, I should add, and hopefully the species we're working on is widespread enough that they should have made it through. But it's hard to tell with species it's so understudied. So thanks for asking about them. So everybody else, if you're bored, go to YouTube and type in velvet worm hunting. I promise you won't regret it. Um, so next question. From a habitat footprint perspective, do you see the ants or moles take up a minor or major coverage of habitat area? Huh. If you had 1 acre, would ants slime it? Wow, I am not good at estimating, so it's kind of hard for me to say. For ants, I mean, what I would say about ants is that it's very rare when we sample environments, we'll do something called a quadrat, which is kind of like a square, and ours are about 30 centimeter by 30 centimeters. If I plunk those down anywhere, it would be weird not to get some ants in there. Ants are just everywhere, and sometimes they're very small species that are less noticeable, but um, ants are basically everywhere. So it's hard to say what proportion would be ants, but I would be shocked if you went outside and picked a random meter by meter square and didn't see hundreds or thousands of ants. Slime molds are trickier because, like I said, when I was talking about the life cycle, the blobby form, you don't see that very often. I've actually only seen them in that plasmodial form in the wild a handful of times, but the tiny spores are literally coating everything outside. So when we want to get wild slime molds, we just go and get a sample of leaf litter or twigs or whatever, and we bring that back to the lab, and we can usually get slime molds off of the under the right conditions. So my guess is that slime mold spores are covering pretty much everything. It's just that the conditions aren't quite right for them to hatch. So yeah, weird, hey? Everywhere. Where do you find meat ant colonies? Meat ants are pretty common pretty much anywhere outside of a city. They tend to like more open woodlands. They're not, you won't find them in really, really densely forested areas with lots of undergrowth. But when you start to get to that kind of mix between forest and grassland, then they tend to be pretty common. I think they're found almost everywhere in Australia, except for maybe the deserts and the rainforest. They're probably not there either, but they're pretty common. They're very noticeable because they build these really beautiful mounds. And the mounds can be, a meter or two across. And the ants will also put little rocks and kind of decorate the mound. And we don't really know why they do that, but it makes them pretty conspicuous when you see them. What sort of pH do slime molds favor? Ooh, that's a good question. From memory, I think they prefer a slightly acidic pH, but don't quote me on that. I have to double check. Yeah, I mean, when we grow them in the lab, we just keep them, we used just distilled water, so just very neutral pH. It's a good question. I'm not sure. Are velvet worms like big tardigrades? No, not really. So they are in the sense that they both, let me think how to explain this. They both belong to different phylums of life. So if you think back to biology, if you took like high school biology, we divide life into different groups. Phylum is a really high level classification. So all the vertebrates, everything with a spine, that's one phylum. Insects, crustaceans, they fall under the arthropods. That's their phylum. So these are really big groups. Velvet worms have their own phylum. So velvet worms are their own thing. Tardigrades also have their own phylum. So each is different from they're each very different from one another. But having said that, both the tardigrades and the velvet worms are thought to potentially be sister groups to the arthropods, which are the group that has insects in them. So not exactly big tardigrades, but similar in the sense that they both have their own phylum. Any examples of what we call kindness in the creatures you've studied? Okay, so there's this cool example that from ants, really. And it's always difficult to use words like kindness because I suppose kindness implies that it's consciously kind, and that's hard to know. But with ants, there are species of ants that go on fights. They battle other ant colonies, which obviously isn't kind. And when they get into these battles, ants in the colony will get injured. Other ants will come and save those ants, like rescue them and bring them back to the colony. And once they're back in the colony, the other ants will look after them, will tend them, will feed them, will lick their wounds. And scientists, not my research group, but other research groups have shown that if the ants are denied that kind of care, that they're much more likely to die, the injured ones. So in a sense, they're rescuing each other, which, it's a kindness. I mean, in some sense, they're saving their own members of their colony, which is probably better for them in the long run. So it gets a bit tricky. But it's interesting. I mean, the other darker side of that is that they have a real cutoff. So if the ants have lost more than 4 of their legs, then they just get left on the battlefield. To the extent that the really badly individuals will actually fight off the rescuers. So it's almost like they're like, too far gone, don't waste your time, go save somebody else. So I probably wouldn't call it kindness, but I think it's interesting because it's a kind of behavior that we wouldn't normally associate with an insect. But next to Do you have any work with culture?
Right. Oh, sorry, one more. I was just going to say we should wrap up in a minute, but I'll let you just answer that last one if you like, Tanya.
Work with culture bearing versus planters. No, unfortunately, I don't really. So my research group hasn't really been studying fire at all until the fires. So I think like a lot of groups, it just wasn't one of our main research questions until we were really caught in that situation. And now we're starting to set up some research to try to look at the impacts of the fires on the velvet worms in particular, but some other insect groups and hopefully the slime molds as well. But that's the first time I've ever really worked in fire. So no, I'm afraid I don't really know much about how bushfire is going to impact things, or at least how they have historically.
Okay, well, look, thank you so much, Tanya. Thanks to everyone for your questions and your attendance. It was a terrific talk. We will, as I said, we're recording, so we'll put this up online as soon as we figure out how. Hopefully it won't be too long. And we'll share that with you. Yeah, thank you so much. We look forward to seeing you again next week for another talk, same time. And then we'll organise some more down the track. Kevin, did you want to add anything before we sign off?
No, remember to register nice and early. And thanks for logging in.
Thank you so much, Tanya. Thanks, everyone.
No worries. Thanks, everyone.